Greetings to everyone and welcome back once again to my educational channel on biology. In this video, we'll be discussing meristematic tissues and growth. So there are two videos on this. In this first video, we are going to talk about the types of meristematic tissues, and that's part 1. And in part 2, the second video, we'll discuss primary and secondary growth. The learning standards for this video are as follows. 1.2 Meristematic tissues and growth. Number 1, after this lesson, we should be able to identify the types and parts of tissues involved in growth. So tissues involved in growth means the meristematic tissues. Alright. Now describe positions of zone of cell division, cell elongation, and cell differentiation in shoots and roots. So as shown here, this is the shoot tip of a plant, and we must be able to identify the 3 zones of growth as stated here. Number 3, identify zone of cell division, zone of cell elongation, and zone of cell differentiation in a seed radicle or root. So seed radicle means the young root of the germinating seed, okay, or germinating plant. So we see here the redwoods, which are the tallest trees in the world, okay, and their growth is due to the activity of meristematic tissues that are active throughout their lifespan. Let's review a little bit of what we have studied in subtopic 1.1. So, plant tissues are divided into meristematic tissues and permanent tissues, right? So we've already discussed permanent tissues, which consists of 3 types. Think of VEG, V-E-G. So permanent tissues consist of vascular tissues, epidermal tissues, and ground tissues, right? And we've discussed all these tissues and their cells. Now, so apart from permanent tissues, there are also the meristematic tissues which are involved in growth. Okay, so these consist of 2 types. Meristematic tissues consist of 2 types of tissues, apical meristem and lateral meristem. So today we're going to discuss this part, okay, meristematic tissues and apical meristem and lateral meristem. Now here's a useful chart of the functions of the various plant tissues, okay. So for meristematic tissues, the function is to carry out cell division by mitosis to produce new cells for growth. Only one function. For permanent tissues, there are 3 types, right? Epidermal tissues, which are needed for protection from injury, absorption of water by the root hair cells, which are epidermal cells. Gut cells are also epidermal cells that allow diffusion of carbon dioxide into the leaf. Now, for ground tissue, remember the chyma, right? The godmother. So you have parenchyma, sclerenchyma, and collenchyma, right? So basically they are used for support, but they support the plant in different ways. And then parenchyma tissue can carry out photosynthesis. Okay, for example, the palisade mesophyll cells, right? They are parenchyma, specialized parenchyma cells. And then parenchyma cells can also store food substances like starch in their cells. Now, for the 3rd type of permanent tissues, the vascular tissues, they are important for the transport of substances in the plant. Okay, they help to transport substances in the plant. Okay, for example, xylem transports water and mineral ions, and phloem transports organic food substances in the plant. Now, here's an overview of the subtopic 1.2 entitled Meristematic Tissues and Growth. This is the first chapter of the Form 5 Biology KSSM syllabus. So in the first video for this subtopic of 1.2, which is this video, we are going to study the meristematic tissues, right? So the meristematic tissues consist of 2 types of meristems, okay? apical meristem and the lateral meristem. So the apical meristem is made up of the shoot apical meristem and the root apical meristem. Okay, so all the meristems are involved in cell division through mitosis to produce new cells for growth. Now, for the lateral meristem, it consists of the vascular cambium and the cork cambium. So what are the differences between these two types of meristem? It will be discussed in this video. Okay, and then we are going to go on to the 3 zones of cell growth in the apical meristem. All right, in the shoot tip and the root tip. Okay, or in the shoot apical meristem and the root apical meristem. So the 3 zones of cell growth are, firstly, the zone of cell division, then the zone of cell elongation, and the zone of cell differentiation. Now, in the second video, which will be the next video, we're going to discuss the 2 types of plant growth, namely primary growth, that is carried out by the apical meristem, and secondary growth, that is carried out by the lateral meristem. Okay, so this will be in the next video. Okay, for this video, we'll concentrate on the meristematic tissues and the 3 zones of cell growth. First of all, let's discuss the definitions of the various types of tissues. So a plant consists of 2 main types of tissues, which are meristematic tissues and permanent tissues. So what's the definition for meristematic tissues? They are tissues that are actively dividing through mitosis to produce new cells for growth. And these tissues are not differentiated. That means if you look at them, they don't have any changes in structure compared to a general cell. They have not undergone changes in structure to become specialized cells. So they look basically like this: rectangular or square-shaped cells with a big nuclei. Each one has a big nucleus in the center, relatively big nucleus, and in some cells there is cell division going on, which is mitosis. So here we see the chromosomes, sister chromatids separating from each other and moving to opposite poles. So what phase of mitosis is this? This is anaphase, all right? And also there may be other stages of mitosis like metaphase, prophase, and all that in the different cells. So location of meristematic tissues, okay, this type of tissues, is at shoot tips, root tips, and in the cambium of the stems. Okay, we'll come to that later in this video. Now, on the other hand, permanent tissues. What's the definition for permanent tissues? They are matured tissues that do not divide anymore, okay, unlike the meristematic tissues. They have undergone differentiation, all right, unlike the meristematic tissues. There are changes in shape and structure of the cells in order for each cell to carry out specific functions more efficiently. Okay, so they have undergone differentiation to become specialized cells. Okay, for example, now this cell is the sieve tube with the sieve plate that has pores. Okay, the sieve plates are the end walls that have pores in them to allow food substances, organic food substances, to pass through them in the cytoplasm. And this, of course, is a hollow cell without cytoplasm, each of these. So these are the xylem vessels, which are also specialized cells, and they have undergone change in shape and structure so that they can transport water more efficiently. So they don't have any end walls and no cytoplasm in the center part here. The cell is hollow and empty to transport water efficiently. So location of permanent tissues are in many places in the stems, roots, leaves, and fruits, right? So today our focus is on the meristematic tissues. Okay, we have already discussed the permanent tissues, but here we try to memorize the definition for meristematic tissues and permanent tissues. So before we go on to study more about meristematic tissues, let's compare the meristematic tissues with the permanent tissues to understand a bit more about the different characteristics of these tissues. So meristematic tissues consist of cells that divide continuously by mitosis, but for permanent tissues, they do not divide anymore. These tissues do not divide. So actually, permanent tissues come from the new cells that are produced by meristematic tissues. Okay, so meristematic tissues actually produce two types of new cells. The first type will continue on with the function of dividing. They are meristematic cells. But there's a group of cells, new cells produced, that will go on to develop into permanent tissues. Okay, so these cells do not divide anymore. All right. In fact, they become fully differentiated. That means they undergo changes in shape and structure in order to carry out their specific functions more efficiently. So when they're fully differentiated, they become specialized cells. Whereas for meristematic tissues, the cells are undifferentiated, as you can see here. Okay, they look like general cells with cytoplasm, nucleus, and cell wall. Okay, right. Then what are the differences in the structure of the cells? Now, for meristematic tissues, they have cells that are small. Each has a relatively large nucleus. Each cell has a relatively large nucleus. Vacuoles are usually absent, and the cell wall is very thin cellulose cell wall here, right? They are like young cells like that. Now, for permanent tissues, cells vary in shape and size. The nucleus is relatively smaller because the cell has elongated after it is formed and after it grows. And then usually there's a large single vacuole in the matured cell for those cells that are alive, right? And the cell wall is thicker. There's a thicker cellulose cell wall for each cell. Sometimes the cell wall is thickened further by lignin or suberin or other substances. All right? So you can see the vacuole in the cells of the permanent tissues, but normally in meristematic tissues, there are no vacuoles, or if there are, they're very tiny. Now, number 7, cells have high metabolic rate, okay, for meristematic tissues or cells. So this is because these meristematic cells are undergoing cell division by mitosis, so they need energy for that. So by having a high metabolic rate, especially a high rate of respiration, they can produce more energy for cell division. More energy through cellular respiration for cell division. Okay. So permanent tissues no longer divide, and these cells have lower metabolic rate. Now, function of meristematic tissues is to enable plants to grow. Okay, and there are two types of growth, that is primary and secondary growth, that the meristematic tissues can carry out. For permanent tissues, they don't divide anymore, so they don't help the plant to grow. Rather, they are involved in protection. For example, epidermal tissues help to protect the plant from injury. Transport, for example, xylem vessels help to transport water and mineral ions from roots to leaves. Photosynthesis, for example, parenchyma cells that are specialized, like palisade mesophyll cells. Okay, they contain chloroplasts to absorb sunlight for photosynthesis and produce organic food substances. Support, for example, sclerenchyma cells here, okay, help to support the plant. Now, number 9, examples of meristematic tissues. There are two main types: the apical meristems at the shoot tip and root tip, and the lateral meristems, such as the vascular cambium and cork cambium, which we will study in this more about in this video. Permanent tissues, what are the examples? Epidermal tissues, parenchyma tissues, collenchyma, sclerenchyma, xylem, and phloem tissues. Okay. Now here you also see a gut cell, which is a specialized cell, specialized epidermal cell. So it also forms part of the permanent tissues. So our focus today is on the meristematic tissues. Now, what are the different types, the two types of meristematic tissues? So meristematic tissues consist of two types based on their location. Okay, we have the apical meristem and the lateral meristem. So apical meristem, the word apical comes from the word apex, meaning the top, okay, or in this case the tip. So apical meristem is located at the shoot tips and all the root tips here. And also the side buds, if the side buds come out from the side of the plant here, the shoot tip does also have the apical meristem. All right. So we can divide the apical meristems into two types. Okay, the SAM and the RAM. What is SAM and RAM? SAM stands for shoot apical meristem because it's found at the shoot tip, the red part here. And RAM stands for root apical meristem, which is found at the root tips, behind the root cap. So this yellow part is the root cap. It is to protect the root apical meristem from injury in the soil. So as it grows downwards, okay, as the root grows downwards, it needs to be protected. The root tip needs to be protected. So this is the yellow part is the root cap to protect the root apical meristem, okay, which is at the root tip. Now, the function of these apical meristems, okay, the shoot apical meristem and the root apical meristem, is to cause primary growth to occur. Now, what is primary growth? Primary growth is the increase in length of the plant to allow the plant to elongate its stem and roots. That's primary growth. Okay, so that means it's like what we call in inverted commas vertical growth, growing upwards. Okay, the shoot growing upwards and the roots growing downwards, vertically. See, so or increase in plant length and height. So root shoots grow vertically upwards in the air, and the roots grow vertically downwards into the soil. Okay, so this is called primary growth. It's the first type of growth that the plant will undergo. As when they have germinated, the plant has germinated from the seed, then it will undergo the primary growth. Just like you go to primary school first, right? And then secondary school. Okay, so there's another growth called secondary growth, which is carried out by this lateral meristem. Okay, so that's it, and the notes will be on the next page, so you can look through it for apical and lateral meristem. So now let's look at lateral meristem. All right, lateral meristem consists of vascular cambium and cork cambium. Right, so they are located in the stem and also the root, and their function is that they're responsible for another type of growth called secondary growth. Okay, so apical meristem is responsible for primary growth, and lateral meristem is responsible for secondary growth. And what is secondary growth? It's the increase in the width or the thickness of the stem, or the diameter of the stem, and also increase in the thickness or diameter of the root. That means the stem will become bigger and bigger, thicker and thicker as the plant grows. That is secondary growth, and it's caused by the lateral meristem. Okay, so what are the lateral meristem? Where are they located? So if we have a cross section, we cut a cross section of a tree stem. All right, a small plant will also have this, or a small tree. So you find that there is a ring of tissues in between the phloem, the blue part here outside, and the yellow part inside, the xylem. This ring of tissues is called the vascular cambium because it's found in the vascular bundle. And then further outwards, near to the external surface of the stem, you can find another type of cambium, also in the shape of a ring. All right, this is the cork cambium that produces cork, which is a substance that can protect the plant, which we'll discuss later. So this is called the cork cambium. It's found in woody plants and in trees, right? Certain plants don't have this cork cambium. So vascular cambium and cork cambium are two types of lateral meristem. Okay, now this is the longitudinal section. So in the longitudinal section, you can see that the vascular cambium, the green one, and the cork cambium runs through the whole length of the tree. All right, to cause the thickening of the stem and the roots. All right, so the growth is sideways, right? The lateral meristem causes growth that's sideways. So that's why we call them lateral meristem. Lateral means side. Whereas for apical meristem, the growth is vertical, vertical growth. So this one is, for apical meristem, the growth is within inverted commas, vertical growth, and that means the lengthening of the plant, all right, the lengthening of the stem, the shoot tip, the lengthening of the stem and the roots. Whereas for lateral meristem, it causes secondary growth, which is, within inverted commas, lateral growth. That means increase in the stem and root diameter and thickness. So these are the notes for what I've discussed just now, okay, on the types of meristematic tissues, okay, based on their location, which are the apical meristem tissues, the SAM and the RAM. Then the function is for primary growth, all right, in which a plant undergoes elongation of stem and roots. And then you have lateral meristem tissues, which consists of vascular cambium and cork cambium, located in stem and root, responsible for secondary growth, meaning the increase in the stem and root diameter or thickness. Okay, so you can copy these notes into your notebook because they are quite complete. So here's an enlarged diagram of a plant to show you the location of the apical meristem. Right, so there's the shoot apical meristem, or SAM, is found at the shoot tip and also at the side buds here. Also shoot tips, but at the sides. Okay, and then that's the SAM, shoot apical meristem. Then there's also the RAM, root apical meristem, is found at the root tips, all the root tips. Okay, the red part here. Now for the other type of meristematic tissue, the lateral meristem, so it's found in the stem, and here we can see the ring of tissues. So the inner ring is the vascular cambium, and the outer ring of tissues is the cork cambium, which is found only in the woody plants that have a longer lifespan. Okay, now here you can see the vascular cambium that runs through the vascular bundles also. It's found in the vascular bundles. Okay, the white ring is the vascular cambium. So our main focus today is on the apical meristems, meaning the shoot apical meristem and the root apical meristem. Okay. Right, so we have discussed apical and lateral meristems briefly. So now let's look at this table of comparison. Okay, so apical meristems are located in shoot tips and root tips, whereas lateral meristems are located in the cambium of stems. Apical meristems consist of the shoot apical meristem, SAM, and the root apical meristem, RAM, whereas lateral meristems consist of vascular cambium and cork cambium in the stem and the root. Okay, apical meristems are found at the shoot tips. The shoot apical meristem is at the shoot tip here and at the side buds, and also the root apical meristems are at the tips of the roots. Right? So apical meristems cause increase in length of shoots and roots, and this is called primary growth because it's the first type of growth experienced by the plant, whereas lateral meristems cause increase in the width of roots and stems. All right, and it's called secondary growth, okay, where the stem becomes thicker and thicker, so there's sort of growth sideways. That's called secondary growth. It's called secondary growth because it occurs later, after primary growth, maybe after the first year or second year of the plant's growth. After primary growth has been going on for a while, then secondary growth may occur to thicken the stem and to give more support to the plant. So apical meristem is responsible for primary growth, or loosely called vertical growth, okay, upwards and downwards. Lateral meristems is responsible for secondary growth, which is sideways growth. The stem will grow thicker and the diameter will become bigger. Okay, so this is sideways. The growth is sideways. So it's called lateral growth. But if you ask to explain what is secondary growth, then you say increase in the width of roots and stems, or in the thickness of roots and stems. Okay, what's primary growth? Increase in the length of roots and shoots. Okay, now apical meristems produce the primary tissues, which we call primary xylem and phloem. So when we say primary xylem and phloem, it means that the Xylem and phloem that's produced during primary growth, that means it's the older xylem and phloem. And then during secondary growth, secondary tissues are produced, which are the secondary xylem and phloem. So these are the younger or the later stage xylem and phloem, formed in the stems, all right? And this produces wood. Secondary xylem will produce wood. So secondary tissues are formed during secondary growth. Okay. Then apical meristems produce new leaves and flowers. For example, at the top here, okay, the new leaves will form and new flowers will form by the side buds. Okay, this is due to the activity of the apical meristem. And then lateral meristems produce woody stem, which we will study in the next video, and the bark of trees. Now, here's a fun fact. Unlike most animals, plants continue to grow throughout their lifespan. Okay, they're always growing. As long as they're living, they are growing. And this is because of the continual division of their meristematic cells or tissues. So the meristematic tissues at the shoot tips and root tips, and also in the stem and roots, they are continually dividing by mitosis. And the meristem never ceases to function and continues to divide throughout plant life. Okay, so here we see the giant sequoia trees in the Sequoia National Park in California, in USA. And these trees, some of them have lived for more, not some of them, they can live for more than 3,000 years. There are trees that are very old, a few thousand years old. Okay, but these ones are maybe the younger ones, but they have lived very long, also many years already, these trees, the giant sequoia trees. Now we're going to talk about the 3 zones of growth in the shoot tip and root tip, which have apical meristems. Okay, so first of all, we have to know the meaning of growth in organisms. This definition is based on your definition in the Form 4 textbook, okay, the last chapter. So what is the meaning of growth and what's the definition of growth? Growth in organisms is an irreversible, permanent process, okay, meaning once the plant has grown, all right, it doesn't, shorten or shrink back again. The process is irreversible, permanent, and it involves an increase. Growth involves an increase in number of cells in the plant and in the organism, size of the cells. So the cells will become larger and larger. The young cells will become larger and larger. Volume of the organism's body will also become more and more, bigger and bigger. Weight of the organism's body. And this is caused by the 2 activities of the cell when they are growing: cell division and cell elongation. All right. So cell division is by mitosis, and cell elongation means the cell enlarges as it grows, becomes longer and longer, bigger and bigger. So growth also involves cell differentiation and specialization. So after the cell has elongated and reached its maturity, it has reached the size at maturity, a specific size, it will undergo cell differentiation, meaning changes in shape and structure of the cell, in order to carry out its function efficiently. So that's the whole thing is called cell specialization. But the change in shape and structure is known as cell differentiation. Okay. Now, in which a cell undergoes changes in shape and structure in order to perform a specific function more efficiently. For example, in the formation of a neuron, which is a very specialized cell, or in the formation of a xylem vessel, okay, which is also a very specialized cell that's important to transport water and mineral ions in the plant. So growth and development involve changes in organisms, starting from the zygote stage until adult stage. So here we see a seed germinating and it's growing. So its root, the radicle is growing, okay, down to the soil. At the same time, the shoot tip is also dividing by mitosis and producing new cells, new leaves. Okay, so the height of the plant begins to increase as this is part of primary growth, which is caused by the apical meristems at the shoot tip and the root tip, where the plant elongates is stems and roots. So let's find out more about the zones of growth. Okay, zones of growth of cells. There are 3 zones of cell growth at the root tip and the shoot tip. All right. So the tips of shoots and roots can be divided into 3 zones of cell growth, which are zone of cell division, usually right at the top of the shoot tip or right at the bottom of the root tip, right? Then you have the zone of cell elongation above the zone of cell division, and then the zone of cell differentiation. So we'll be discussing this afterwards. The growth that occurs in these zones is the primary growth in plants. Okay, when the growth occurs, the shoot will increase in length. Okay, it will grow higher, and the root will increase in length, so it grow further down to the soil. That is primary growth, or what we call loosely vertical growth. Right, so in this diagram, you can see the 3 zones of cell growth at a shoot tip. By shoot tip, we mean the young bud of the shoot here, right? The very top of the plant. It can also grow out from the sides between a leaf and the stem, right? Right? So all these side buds are called axillary buds. They are young, very young tissues that can develop into leaves and branches and so forth. Right? So they also contain the meristematic tissues, apart from the one at the top. Okay, let's say we take this top tissue here, right? The shoot tip here. So we can see the 3 zones of cell growth, right? Starting with the very first part at the top, the zone of cell division, which contains the shoot apical meristem. Then we have the zone of cell elongation below, and then further down, the zone of cell differentiation, right? So in the zone of cell division, we can see the very top of it contains, the zone of cell division contains the shoot apical meristem or SPM. Now, leaf primordium means a group of cells that will develop into a new leaf as the shoot grows, right? All right, so here in the zone of cell elongation, vascular tissues start to form. So the cells are growing longer and bigger. Okay, from young cells they become bigger and bigger. And finally, in the zone of cell differentiation, you can see specialized cells that have undergone differentiation. All right, so there's the xylem tissue and the phloem tissue. Okay, so now we're going to discuss the cells, the structure of the cells in these 3 zones in more detail. Right, the 3 zones of cell growth, all right, which are the zones of cell division, cell elongation, and cell differentiation. Let's start with the zone of cell division, which contains the shoot apical meristem. How do the cells look like at the shoot apical meristem, in the shoot apical meristem? All right, so the cells are small and they may have some small vacuoles. The nucleus is relatively large, okay, and the cells are packed tightly together, very closely to each other, and it has a thin cell wall. Each cell has a thin cell wall, okay, and dense cytoplasm. So we can see the cells in various stages of mitosis. For example, what phase is this? Where the chromosomes are all at the center of the cell, or at the metaphase plate or equator of the cell. Now this is metaphase of mitosis. This stage is metaphase of mitosis. Here may be prophase, and this one is anaphase. You see, so you can see the cells at different stages of mitosis, or they may just be, resting in the resting phase, all right, after cell division, or when the 2 new cells are formed. So you see each cell has a large nucleus, relatively large nucleus compared to the whole cell. Now, that is how we identify the zone of cell division. Small cells like this, with small cells like this. Now, in the zone of cell elongation, the cells are becoming larger and elongating, right? So the vacuoles, small vacuoles start to form, and then they combine together, so the vacuoles are enlarged. And finally, there will be only one big vacuole when the cell is more mature. Okay, all the vacuoles will combine to form one large vacuole, and the cell has, you can see here that this cell has elongated. Then the cell wall is thicker than that in the shoot apical meristem. Now, in the zone of cell differentiation, it's quite easy to identify this zone because we can see a lot of specialized cells, okay, where there have been changes in shape and structure so that the cells can carry out their functions efficiently. So what type of cells are these? All these three. Yes, they are xylem vessels. Not xylem cells. You have to use the word vessels. Xylem vessels. And this one, so these two cells, sieve tube and companion cell, is part of the phloem tissue. Phloem tissue. So this one is the sieve tube. Okay, the blue one, with the pores in the sieve plates. Okay, at the end walls. These are the end walls, and they are called sieve plates because they have pores in them. Now, secondly, this diagram shows three zones of cell growth at the root tip. Just now was shoot tip, so now it's root tip. So for roots, it's a bit different because there is a root cap at the end of the root tip, okay, to protect the meristematic tissues from injury as the root is growing downwards into the soil, in between the soil particles. So this root cap prevents the root apical meristem from being damaged. What happens if the root apical meristem is damaged? If it's damaged and the cells die, then there are no more cells for cell division, right? So the root will stop growing. All right. Now, so above the root cap, we see the root apical meristem, all right, in the zone of cell division. And the cells here are similar to the cells for the shoot apical meristem. They are small, look a bit square, okay, in shape, or rectangular a bit, and they have large nuclei, thin cell walls, small vacuoles, and then cytoplasm. So if you look at the cell under the microscope, you may be able, if you look under the microscope and see the cells of the root apical meristem, you may be able to observe the cells undergoing some stages of mitosis, like in this picture here. All right, because this is the zone of cell division. Right, the root apical meristem carries out cell division. Now, next is the zone of cell elongation, which is above the zone of cell division. So for the root, we start from the bottom, and then we go upwards to find the other two zones after the zone of cell division. For the shoot, we start from the top, and then we go downwards from the root, from the zone of cell division, then to the zone of cell elongation, and then zone of cell differentiation below it. But for the root, we have to go upwards. So the second zone is the zone of cell elongation. Here we see the cells elongating, and the size of the cells will become bigger and bigger. At the end of it, we may get a matured, we'll get a mature cell, but one that hasn't undergone differentiation, all right, and it has a big central vacuole. So more about this later. And then in the third zone, which is the zone of cell differentiation, we can see cells that have already differentiated, like to form the xylem vessels, for example, xylem vessels here. And for these ones, you can see the root hair projecting out. So these are the root hair cells. With the root hairs to provide a larger surface area for absorption of water. Right, so now for each zone of cell growth, we have to be able to explain the characteristics of the cells found there, right? So firstly, the zone of cell division, which is at the root tip and shoot tip. Location, the zone of cell division is located at shoot tip and root tip of a plant, and it includes the apical meristems, SAM, which is the shoot apical meristem, and RAM, meaning the root apical meristem. Of course, in exam, you cannot write SAM and RAM. You have to write out in full the name, shoot apical meristem, root apical meristem, right? Which consists of actively dividing cells, meristematic cells, that undergo mitosis. And the function is to produce new cells for growth, right? So the location is at the shoot tip right at the top, for example here, or the root tip, right? The apical meristem is the red colored structure here, right? So inside these apical meristems, you will find cells that look like this. The cells in the apical meristems are small cells, right? And they have thin cell walls, large nuclei, as you can see here, no vacuoles inside, or if there are, the vacuoles are very small. The cytoplasm is dense, and the cells are tightly packed together, okay, with no intercellular air spaces, lack of intercellular air spaces. So the increase in the number of cells causes elongation of the plant stem or of the roots. All right. So this is primary growth. All right, and it happens throughout the life of the plant, starting from the time when the seed germinates, and the young plant starts to grow. Okay, there's already there's the mitosis will be carried out at the meristematic tissues in the zone of cell division to produce new cells for growth. Okay, so the increase in the number of cells will cause the root tip and the shoot tip, to elongate, or to grow longer. For the shoot, will grow higher up, and the root will grow further down into the soil. When the new cells are forming, the cells formed previously are then pushed to the next zone, that is the zone of cell elongation. So these will be cells that are going to differentiate later on to become permanent tissues. Okay, but there are some new cells formed that will maintain or retain the ability to divide. They will still be meristematic tissues or meristematic cells. Okay, whereas other cells may undergo cell differentiation later on. So these cells will be pushed into the zone of cell elongation, the next zone of growth. The second zone of cell growth is the zone of cell elongation. Okay, so the zone of cell elongation is found below the zone of cell division at the shoot tip. All right, and it's the place where the cells, the young cells, are increasing in size, are elongating and enlarging. For the root tip, the zone of cell elongation is above the meristematic tissues and above the zone of cell division. All right. So the zone of cell elongation consists of young cells that are increasing in size. All right, like here, like what we see here. The increase in size of cells occurs through, number one, diffusion of water by osmosis, okay, whereby the water diffuses into the cell by osmosis, and then it is stored in the vacuoles. So the cell will have more and more vacuoles, and these vacuoles will become larger and larger, and some of these vacuoles will join together, fuse together to become bigger and bigger, right? Now, apart from diffusion of water into the cell by osmosis to enlarge the cells, there's also absorption of nutrients into the cells, all right, to produce new cytoplasm also, all right, to produce new cytoplasm. Right, then how does the cell expand in size? How does it elongate? Through a process known as vacuolation, all right? So in vacuolation, as the water diffuses into the cells, the vacuoles form and then they fuse together to form larger and larger vacuoles. And then these vacuoles, the water in the vacuoles will exert pressure on the cell walls. Okay? And then probably because the cell wall is not thick yet, okay, at the beginning the cell wall is thin, so the cell wall can expand to a certain extent. So as the water in the vacuole exerts pressure on the cell walls, this will push, elongate, and widen the cell. All right. Now, at the same time, the small vacuoles will combine to form a large central vacuum. Alright, so in that way, cell elongation occurs. So after the zone of cell elongation, we have the zone of cell differentiation. It's the third zone of cell growth. Now, in the shoot tip, where is it located? You have to go from the top downwards to the third zone. Alright, so it is the zone here, after the zone of cell elongation, that where we can see the differentiated tissues that have differentiated, such as xylem tissues and phloem tissues. Alright, that shows us that this is the zone of cell differentiation. For the root, we have to move from the bottom upwards, right? So after the zone of cell division here, with the zone of cell elongation, and then the zone of cell differentiation. And in this zone, the zone of cell differentiation in the root, we can see root hairs extending out from the epidermis, because the root hair cell is a cell that has undergone differentiation, right? Okay, so let's define differentiation. Differentiation is a process whereby cells change in shape and structure to become specialized cells that carry out specific functions efficiently. All right, so this is a very good definition, and you can copy it down and memorize it. For structured and essay questions, you may have to explain this way. So, process whereby cells change in shape and structure. All right, for example, here we have a generalized cell with just the simple structures, all right, the normal plant structures. Okay, and when it has matured, it will undergo differentiation to form a specialized cell, right? So in differentiation, for example, to form the xylem vessels here, right, the cytoplasm of the cell will disappear, leaving an empty hollow center here, right, for water to be transported easily without obstruction inside the cell. Okay, and then its cell wall will be thickened with lignin, okay, to provide strength to the cell wall, so that the xylem vessel will not collapse when it is transporting water, right? So these are some of the changes in shape and structure that occur so that the xylem vessel becomes a specialized cell with special shape and structure to carry out its specific function efficiently. All right. Same for sieve tube. The sieve tube is a cell that has a sieve plate with pores to allow the organic food substances to be transported by the cytoplasm from one cell to the next. Right. So the zone of cell differentiation, okay, here and here, consists of differentiating cells that differentiate once they have reached maximum size. And once they have reached the maximum size, like this cell here. Cells differentiate to form permanent tissues, such as epidermal tissues, xylem tissues, phloem tissues. Okay, that contain these specialized cells. Okay, like xylem tissues have xylem vessels, and phloem tissues have sieve tubes and companion cells, which are specialized cells. Now, number 4, 2 examples of cell differentiation, okay, that leads to cell specialization. So, epidermal cells in leaves differentiate to form gut cells with a bean shape, right? And these gut cells have chloroplasts. So, the structure of the cell, the bean-shaped structure, with the thick inner wall and thin outer wall, and the chloroplast, all help the gut cells to carry out their function of regulating or controlling the opening of the stoma, which is the leaf pore, so that carbon dioxide can diffuse into the leaf for what? For photosynthesis. And another example are the epidermal cells in the roots. They differentiate and form the root hair cells, which have long projections to increase the surface area of the root for faster absorption of water from the soil. All right, so that is cell differentiation. Right, so that's all for this lesson. In the next video, we are going to talk about primary and secondary growth. Okay, so thanks for watching. So the notes are very good and you can copy them into your notebook. All right, and thanks for watching and take care. Do share, like, and subscribe. Till we meet again, goodbye.